India is a repository of biodiversity. Gigantic trees, tiny lichens and mosses, little ferns, exotic flowering plants. Endless is our plant diversity. India has also been home to a variety of plant species that were introduced several centuries ago as commercial crops. Many of these are welcome guests since they continue to contribute immensely to the Indian economy. But there are some guests who have stealthily entered India and have become aggressive invaders. With impunity, these invaders have colonized large tracts of unused land and plantations. These have now begun to invade the virgin forests too. These are weeds, the biological invaders. Of more than 3 lakh plant species known to the world, hardly 3,000 are of any use to us. While all unwanted plants are not weeds, there are quite a few plants that qualify to be called weed, for they pose a threat to the survival of useful plants. Some such plants are weeds because they're responsible for many a human or animal health hazard.
Thus, a weed is a troublesome plant which grows intentionally in areas where it is not desired. The most important exotic weeds in Kerala state are Lantana, Chromalina or Eupatorium, Parthenium, Mimosa invisa, and Mycenia micrantha. Of these weeds, Mimosa invisa and Mycenia micrantha are recent introductions. They are also the most dreadful because they pose serious threat to our native plants, plantations, and agricultural crops.
Mycenia micrantha is a member of the Asteraceae family. It is a versatile climber. It has ovate deltoid leaves with serrate margins. With its supple yet strong stem, it winds the host plant to colonize and thrive. It is a native of tropical and subtropical zones of South America. It may have reached Kerala along with wood or seeds or fodder grass imported from countries like Brazil and Costa Rica. If this is true, the Cochin port may have been its embarkation site. In the far northeastern states, Mycenia micrantha has been in existence since the early 1940s, apparently introduced as a non-leguminous ground cover for crops. In Kerala, the luxuriant growth of this weed along the road and rail lines suggests that it could have spread there through these channels of transport. Perhaps for this reason, Mycenia is the only vegetation in many places close to the Cochin railway station and the shipyard. Deforestation that opens the canopy of the forest has created the most favorable condition for the spread of Mycenia. It has now covered large tracts of forest plantations and fringes of prime forest in central and south regions of the state, besides colonizing areas adjacent to water bodies. Observations in Kerala show that Mycania infestation was high in the low-lying areas below 500 meters mean sea level. In regions above 1000 meters mean sea level, Mycania infestation was minimal. Studies conducted by the scientists of the Kerala Forest Research Institute at Pichi have thrown more light on the potential threat that Mycania micrantha is. This perennial weed grows vigorously during the monsoons. By August, the weed blooms. Fruit setting occurs after 17 to 21 days of flowering. A single plant yields as many as 30,000 seeds with a viability of 30%. Seed dispersal is during the months of October and April. The high seed viability helps them proliferate quickly over vast areas. Besides sexual reproduction, Mycania is capable of vegetative reproduction too. New saplings sprout from the nodal region and from the rootstock. This dual capability makes its progeny multiply quickly and certainly. As many as 30 to 40 plants have been observed to sprout up from the suckers of a single plant. Together, these plants can cover an area of about 20 to 25 square meters within a short span of time. These dreaded plants grow at an estimated rate of 8 to 12 centimeters a day, which is comparable to the growth rate of bamboo plants. Though not a parasite, Mycania destroys supporting plants by smothering them, reducing light penetration and leading to the tent house effect. By consuming a large share of water and other nutrients, these plants stunt the growth of other plants.
Besides these, Mycania can cause harm to native plants and their crops through production of phenolic and flavonoid substances which are allelopathic. These allelopathic substances can retard growth of native plants and inhibit germination of seeds.
That these weeds retard the growth of useful plants is in evidence in the teak plantations in the Varachal forest range of Kerala. Many eucalyptus plantations in the state are also under the threat of Mycania. In some locations, Mycania has subsumed trees, often reaching a height of over 30 feet. Slowly, but dangerously enough, this alien weed is making its presence felt in many forests of the state. Deforestation has made it easy for the encroacher to further its aggression. It won't be long before it reaches the evergreen forests. The unchecked growth of Mycania has reduced the yield of forest produce. The reed collectors complain of the reduced size of the reeds and the difficulty in speedy collection. Further, in many places, the overgrown creepers are found to be blocking the path of wild animals, leading to their migration to other places. Mycania menace has also affected plantation crops such as coffee, pepper, banana, rubber and coconut trees, often reducing the yield. Weeding out Mycania is cumbersome and expensive. This has begun to tell upon the cost of cultivation. To control the threat of fast-growing Mycania, KFRI scientists have evolved a strategy.
Several methods, including mechanical, chemical and biological, have been attempted in India and elsewhere to control Mycania, but no single technique was found effective in controlling the weed. Studies at KFRI have shown that some of the commonly available herbicides are useful in controlling Mycania, but this control is short term because after a few months' time the weed will regrow. It thus appears that biological control using natural enemies of the weed is the best option to control Mycania.
The mechanical method is found to be effective in small land holdings, but in large plantations and in natural forests, mechanical method is not cost effective. Researchers have investigated the use of chemical herbicides. Of the 5 herbicides tested, glyphosate applied at the rate of 5 liters per hectare was found effective in controlling the Mycania for a short period. New saplings were found sprouting in the herbicide sprayed area soon after the monsoon. Being highly toxic, herbicides cannot be continuously used to eradicate Mycania. The third is the biocontrol method using pathogenic fungi. KFRI scientists have isolated 9 pathogenic fungi from the leaves of the Mycania plants. The scientists thought that these cultured fungi, when released, would induce the leaf spot disease in Mycania plants. But none of the pathogens so developed was able to cause any serious damage to the hardy Mycania. Hence, the scientists have now sought to use certain biocontrol agents from their native range to check the surge of Mycania.
Colleagues at Cabby Bioscience UK, who have collaborated with this project, have identified a rust pathogen by the name Parthenium spegasini from the neotropics, which has the potential to control Mycania. Attempts are currently being made to import this pathogen to India to try its potential in North and South India.
This host-specific fungus from Brazil causes severe damage to the stem and leaves of Mycania. This pathogen is slated to be tested for host specificity at the National Bureau of Plant Genetic Research at New Delhi before releasing it in Assam and Kerala. While the battle against Mycania continues, there are threats from other invaders too. One such invader is Mimosa invisa, which has already established its dominating presence throughout the state. How then are we going to tackle these weeds that pose a serious threat to the already ravaged fragile ecosystem of the country?